Microscopy module walkthrough. So this week you click on simple microscopy. As usually, you have a module overview, then to know, to see, to do, and then optional materials. Under to do, you find the assignments, the quiz, the question set, and the light microscopy. But let's talk about how this all goes together. So in this lab, we're going to look at compound microscopes, how they work. And then we're going to look at some cells and we're going to look at how they change with environmental impacts. What you're going to do is you're going to learn how to use the microscope. And you can read the rest of this yourself, but this tells you what all you're going to do this week. So read through this. Then this is what you're going to have to, what you're going to be doing. Read the lab handout, watch the lecture, complete the lecture quiz, watch the demo activities. If you're struggling with the lab activities, watch the demo, and then complete the question set and the handout. Click on the next page. Here's your lab handout, and I'm just going to run through the lab handout real quick. As usually, you've got those 3 items that we always have. And in the lab handout, you see the microscopy activity. When you click on it, it tells you all the things that you're going to do. And that's what I'm going to go over real quick. So it goes about, we're going to look at the equipment, the microscope, how it works, why it works the way it does. And then we're going to look at a couple of specimens and we're going to play with those a little bit. So the first things, we're going to talk about the function and the items, the names of everything, what they do, and I'm going to expect you to be able to identify those. And there's a picture, and use that picture to answer the questions in the exam. Then if you're face-to-face, one of the things you want to pay attention to is how to put a slide into a slide holder. Most students struggle with that. That's really something you can only experience when you're face-to-face. But pay attention to that. And then in the lab, if you're online, you're going to do this with just pictures. In the lab, you're going to actually do this with a microscope. And this is one of those that translates very badly online. So if there's at any way possible that you can be there for this lab, be here for this lab. Because there's nothing quite like looking through a real microscope and trying to figure out how to focus and how the pieces go together. Because we're playing with functions of the microscope. And the only way you really can get those across is by really experiencing it. So we're going to look at the hydra first, which is relatively big, then a paramecium, which is a little smaller. And then I'm going to start playing with characteristics of the microscope. We're going to make a wet mount of a letter E. And then you're going to look at that letter E under the microscope and see what happens when you move it. And then here's where it gets complicated. We're going to start measuring things with the microscope. So this is one of those where you're probably going to be stuck when you're trying to do this yourself, and there's lots of help available for that. And then last but not least, we're going to do the impact of salt on allodia. So what changes, this has to do with osmosis. So what happens when you change the salt concentration on the inside, on the outside. And then you have the question set. As usually, you have the question set available online, same questions as here. And let's just kind of go through this. You have this question set, parts of the microscope, the letter E, how does it move? The depth of field, the color thread. We're going to try to see which piece of thread is on top and which is on bottom. And again, this is one of those that's much better done in the lab than it is online, because you can't really experience this online. And then measuring with a compound microscope. How can you measure the size of a paramecium using a microscope? We're going to go over that in the lab. So there's lots of help if you're struggling with math, but if you're going to be there in the lab, that'll be helpful because you can experience kind of what's happening. Then microscopic features of leaves. We're going to look at these little plants, which are beautiful. Their little stomata are green against a purple background. And then we're going to calculate how many of these stomata, how many stomata we're going to have in the size of a quarter. And again, math, we're going to go over this in the lab. And then last but not least, Elodea. Again, this is hard to see online. You just see a still picture. Experiencing it face to face is really going to be a much better idea. So hope to see you in the lab if you haven't gathered that part yet. But so that's the lab handout. There's the lecture. So I talk about all the things that you need to know. If you're struggling with what I'm talking about in the lab, preview this lecture. The information doesn't really change. My lecture may change slightly, but it doesn't really change that much. Go on to the demonstrations. All of these demonstrations, we're going to go through all of this in the lab. So for those of you who are on face-to-face, this is just going to be a review, or you can do it in advance and then, see if it really looks that way, if that helps you. This is how to move a microscope, how to work with it. If you want to look at it in advance, go look at it in advance. You've got all these things available to you. Preparing a wet mount, that's how you're going to do this letter E thing. This is a microscope. It's really not the same thing as a real microscope. It's second best. It's not as good. And so we're attempting to duplicate things here. If you're in the face-to-face class, this is good. This is good review. Otherwise, this may be a little troublesome. And then you've got the assignments, which are the same as usually. You've got a quiz. You've got the question set, which are the questions. And then you've got the Labster assignment. So the quiz, handout question set, Labster assignment. And optional materials is the story of Antony van Leeuwenhoek, which is an amazing story. The first guy who ever saw life under the microscope. And no assignment associated with it, but it's an amazing story, and I hope you get a chance to see it. That's all I got.